Mitosis is a type of cell division in which one cell divides, to produce two new cells that are genetically identical to itself. Mitosis is a way of making more cells, which helps the organisms to grow in size. Mitosis also helps in repairing the damaged tissue or replacing the worn-out cells. Single-celled eukaryotes like yeast, meiotic divisions are actually a form of reproduction, adding new individuals to the population. The processes occurring during mitosis have been divided into different stages. Prophase, Metaphase, Anaphase, Telophase. Before entering mitosis, a cell spends a period of its growth under interphase. It undergoes the following phases. G1 phase. This is the growth period before the synthesis of DNA. S phase. This is the phase during which DNA synthesis takes place. G2 phase. This is the growth phase between the end of DNA synthesis and the beginning of the prophase. Prophase immediately follows the S and G2 phases of the cycle and is marked by condensation of the genetic material to form compact meiotic chromosomes. Chromosomes are composed of two chromatids attached at the center called centromere. The meiotic spindle begins to form. The spindle grows between the centrosomes as they move apart. The nuclear envelope starts disintegrating. At metaphase stage, the microtubules start pulling the chromosomes with equal force and the chromosome ends up in the middle of the cell. This region is known as the metaphase plate. At anaphase, the splitting of the sister chromatids occurs. The chromosomes are then pulled towards the pole. The centromere of each chromosome leads at the edge while the arms trail behind it. In telophase, the meiotic spindle is broken down into its building blocks. Two new nuclei form, one for each set of chromosomes. Nuclear membranes reappear. Cytokinesis, which is division of the cell contents, takes place. The chromosomes begin to decondense and return to their stringy form. When cytokinesis finishes, we end up with two new cells, each with a complete set of chromosomes identical to those of the mother cell.